today. Um, so, first of all, one of the things that I love about this paper is it gives you very brief, right, but a little bit of history of antibiotic resistance. All right. And so we'll go with Olivia. How long has antibiotic resistance been around? Is this a new thing? And what are some ways that we kind of know that or think that it's
I don't remember when I was reading. I think it said that it wasn't a new thing, and it's something that's been just like going on for a while because we've had to continuously change antibiotics because they've been resisting. So
okay. Can someone add to that maybe a little bit or remember a part of the paper?
I remember it said that anti-resistant bacteria is like ancient, just like antibiotics, and there's evidence that identify different R or ARGs in like permafrost samples.
Right, perfect. Right. So we know we have discovered bacteria that are buried deep in permafrost. We've found bacteria that are in caves that haven't been touched or visited in a long time, right? Thousands of years. And so even though there might not have been a pressure or a need for those antibiotic resistant genes to be present, they clearly have been in those bacteria for a long time. And there's even a point here where it talks about a beta lactamase, and we'll talk about that a little bit later. But beta lactams, right? So penicillin being one of the big ones that you guys have all probably heard of. Some bacteria will produce something called a beta lactamase, which is an enzyme, right, that neutralizes penicillin. And so they talk about here, right, transfer between bacterial species millions of years ago. So the fact that bacteria, we think, for millions of years have been able to share this information, right? So the idea of antibiotic resistance is not new. And one of the things that we'll kind of touch on a little bit later, and maybe when our distinguished guest lecturer for Summer Thursday, Professor Perry, talks about fungi a little bit, she might mention, right, that fungi are a great source of antibiotics, as well as other bacteria. And so even before we were kind of around and doing the things we do, right, there was competition amongst fungi and competition amongst bacteria. And they would produce their own antibiotics, right, which is where we get a lot of our drugs from, or derivatives of our drugs. And so they would have to develop resistance against other bacteria and against other fungi, right? And so this is not a new thing. Now, kind of to Olivia's point that she was hinting at, right, is this idea of selective pressure through human antibiotic use. All right. So what are we doing, right? And please don't overthink this, right? Megan, I'll give you this one. What are we doing that's increasing the presence or the prevalence of these antibiotic resistant genes in bacteria?
We're using a lot more antibiotics, like in stock feed or just in cases where we might not need to.
Perfect, right? So overuse. And so the data in this paper, this is a 2016 paper, right? And they cite data, it was 2000 to 2010. There was an increase by 36% of antibiotic use. I can't remember if this is global, right? And so, right, there's been a huge increase. And Megan's right, we'll talk about it's part of food sources, right? Antibiotics actually in chickens increase the growth rate in chickens, so they get injected. One of the reasons that a lot of your meat products are so affordable is we can keep animals in close proximity and close quarter. And so one of the things that we do to keep them healthy when they're jammed, packed together and walking in each other's feces, right, is we give them a bunch of antibiotics. What is one of the number one causes of antibiotics prescribed in the United States? Does anyone know? Megan, I can't read your lips. What did you say?
UTI.
Well, okay. So I should have read this question a little differently, but what I'm getting at is that actually we see a huge an uptick in antibiotic prescriptions during flu season, right? What is the flu? What is it? It's a virus, right? Antibiotics don't do things to viruses, and so there's a lot of misuse of antibiotics, right? And that puts selective pressure on bacteria because they don't want to die, just like you probably don't want to die, right? And so they adapt and they change. Now, one of the things that I'm going to want you to be really comfortable with, right, are, I would say, kind of the three classic modes of horizontal gene transfer: conjugation, transformation, and transduction. All right, and again, we'll visit these again in lecture, but this paper is kind of a great source along with your textbook for you guys to review these different kind of mechanisms, right? And one of the things that I want you to appreciate is that they can lead to or accomplish the same thing, but the mechanisms are very, very different. So before we go on, right, and leave this paper, because we're going to revisit these topics, let's see. Amanda, can you briefly explain the concept of transformation? What's happening in transformation? How is it different from the other types of horizontal gene transfer?
So is transformation when one bacteria is giving DNA to another bacteria?
Yes, but how?
Is that when they're using their pili? Or is that conjugation? I think that's conjugation. Okay, I'm not sure about transformation then.
Can someone help Amanda with transformation?
Transformation is when there's DNA in the environment around the bacteria and they uptake it into their cell and integrate it into their DNA.
Okay, perfect, right? So this idea of free-floating, right, DNA, right, that a bacterial cell can take up. Now, I mentioned this in your paper. What does bacteria need to take up DNA, or what causes it perhaps to take up DNA? Right, so the idea would be if I took a bucket full of, Allie, what's your favorite fruit or vegetable?
I like watermelon.
Watermelon, all right, perfect, right? If I took a bucket full of watermelon DNA and I dumped it all over Allie, would she incorporate watermelon DNA into her cells? Correct, she would not, all right? So why can some bacteria, or what do bacteria do in order to be able to incorporate that DNA?
I think it says something about competence in the paper, that the bacteria has to be competent and the DNA has to be stable.
Okay, perfect, right? So I love that, right? So what I'm looking for, the cells have to be competent, right? They have to be willing to take up DNA. Not all bacteria cells can do this, right? And if you actually go back and look through your paper, right, you'll see that receptors are required for this, right? So we have receptors, or bacteria have receptors that can bind this free-floating DNA and then help bring it into the cell, right? And a lot of times, right, it's some type of selective pressure. So this paper focuses a little bit more on antibiotic resistance. And so if I'll give Elena, I start giving her a bunch of antibiotics because she's sick, right? It starts killing bacteria, all right? So now we have free-floating bacterial DNA around, and the bacteria that are surviving or hanging on, right? They feel the pressure or the stress from those antibiotics, so they then become competent, right? So for the most part, a competent cell has to be induced. There has to be some type of environmental stress. Could be antibiotics, could be something else, right? And then basically it becomes a free fall or free for all, right? We're just trying to bring in or grab genetic information from our fallen compadres to see if they have some piece of information that can be useful there. All right, perfect. I think, who is it that I asked? I think Amanda kind of explained conjugation relatively well, right? So that's the sex pili or the fertility pilus. We'll talk more about that. One of the things I want you to note there and kind of file this away is that plasmids are typically what is transferred during conjugation. It's possible for a whole chromosome to be transferred. We'll talk about why that's not likely in just a little bit. Lauren, did you have a question?
So if they're trying to take up antibiotic resistance genes, but they're taking them up from cells that have died from antibiotics, how do they get them? Because they are the ones that died.
Right. So I guess the best way to describe that is you can say there's various levels of antibiotic resistance, right? So, you know, what would happen is, like, let's say you have an infection, you're ill, you're not feeling well, and the doctor prescribes you antibiotics for 10 days. And day 5, you start feeling better, right? So you go off of the antibiotics. Well, in those first 5 days, you've killed the bacteria that are most susceptible. And that's probably a very large population of them. The bacteria that are left, you know, are resistant in a sense. If you would have finished your 5 days of antibiotics, right? The way I kind of look at it, or you can kind of think about it, is an antibiotic is punching a bacteria. All right. And so the first 5 days, right? You know, you go and you beat up all the little weak bacteria. Day 5 to 10, then you're focusing on the stronger bacteria. And if you punch those stronger bacteria for 5 extra days in a row, 10 days total, you take care of them. All right. But if you don't finish it off, then those bacteria that are somewhat resistant, right, start to multiply, and that information can be shared with other bacteria. Does that kind of make sense? And so resistance is not invincibility, right? You could, well, in some cases it's not invincibility, right? You've heard of some of the bacteria that's resistant to every drug, but right, for the most part, resistance is referring to a decreased susceptibility. And so if you were to follow the doctor's orders and finish the prescription, a lot of times you would knock off those bacteria that are even, you know, somewhat resistant. So does that help a little bit? Kind of makes sense. Perfect. All right. The last one that I want to talk about, and I'll have Rachel Dickey explain this one to me, is what's the basic concept of transduction?
Isn't that where like bacteriophages are what transfer the DNA to something else?
Okay, perfect. And what's a bacteriophage? You know?
I know what it looks like in the picture.
Okay, cool. What does it look like?
It has like things that like can like grab on.
Mm-hmm.
And it has the DNA like in the top part of it.
Mm-hmm. So those of you that are interested in outer space, which I don't know why you would be, but maybe you are. If you've ever seen pictures of the lunar lander, you can go ahead and look that up and Google it if you don't believe me, but it kind of looks like a spaceship type thing that we use to land on the moon. So bacteriophage is actually, right, a virus that is specific for attacking bacteria. So Anna, what do viruses do to cells? How do they work?
I mean, they attack them, certain parts of the cell, and
Okay, so
invade it and like destroy it, kind of.
Yeah, they destroy it, right? And the reason that a lot of them destroy it, and we'll talk about some exceptions, is because viruses can't do things on their own. So they infect a cell, they take over the cell, and they use that cell to make a bunch of copies of themselves, right? And then they assemble, and then a lot of times the cell will lyse and they'll burst out. For bacteriophages, sometimes what happens, right, is when they make copies of themselves, and they are repackaging, right, making new viruses to get ready to go out, bacterial DNA accidentally gets incorporated, right, into the shell of the virus. And so basically you have a viral shell with bacterial DNA in it, right? And sometimes that bacterial DNA contains really valuable information, all right? And so in this case, even going to kind of Lauren's, question, why would you want stuff from dead bacteria? Well, right, a virus is not an antibiotic. And so even though, right, in this case, that virus killed the bacteria, it still may have information that allows other bacteria to be resistant to drugs, right? So there's a lot of different things that are kind of going on, right? So this is a viral infection, right, that actually ends up passing along important information to bacteria. So when that bacteriophage, that virus, attaches to a new bacteria, it injects bacterial DNA in, and that's then incorporated into the chromosome. And we'll briefly mention this in, I think, the viral unit is when we talk about it. But some of the pathogenic bacteria that you guys may have heard of are actually pathogenic because of previous viral infections at one point in their history many years ago. All right. So horizontal gene transfer, we're going to talk about this more. So love it, learn it, live it. I don't want to take too much more time on this, but this is kind of a cool, I think, paper emphasizing the symbiotic relationship between plants and fungi. So who can just give me a very brief description of what happened in this paper, or what they're looking at, or why we should care? Laura, do you remember what happened in this paper?
I'm not sure about the answer to your question, though. I did read it.
Okay, well, tell me something about the paper.
I was talking about like animals transferring the cells and them growing in different places, but I'm not sure about specifically the fungi and the plants together.
Okay. That's an important part, right? One of the things they're focusing on, right? And this is really interesting, right? Because it's basically showing that two invasive species are relying on one another. All right. And so Laura mentioned the animals, specifically from your quiz, right? The wild boar, right? And there are also some wild deer, right? And both of these have been introduced to this. This is a small island, I believe, off the coast of Argentina. All right. So how does it work? What's the basic premise? Who can help me out there? So we have two invasive species. How are they working together?
The species are, I think, obligate mutualists.
Okay. Mm-hmm.
Okay, and they so the tree seeds can like spread on their own through the air, but the fungi that the tree needs to survive can't be spread on its own through the air. So the wild boar and deer will eat it and then produce fecal matter. And if that ends up in the same area as the tree seeds, then the tree seeds can grow.
Okay. So that's a pretty good summary, right? And so in this paper, right, and it's different, right? They call it a tree plantation, right? So we would call it a tree farm. And the trees on this tree farm, right, are non-native to this island, all right? And the fungi that they need doesn't really exist outside of the tree farm. And so if seeds spread to other parts of the island, it didn't used to be an issue, right? But now you have these animal populations, right, that are also non-native that happen to have the fungi that they need, right? So boars actually like to eat fungi, right? They eat fungi, they poop, right? Seeds find poop or soil that was near the poop, right? And then it forms this mutualistic benefit. And so now what they've started to see is that these islands, or not these islands, these trees are popping up on different areas of the island off of plantation because the animal species, the boar and even some deer, are spreading this fungi so that the trees can also spread. And, one of the things that, I'm not sure how much we'll touch on it, but I'll be sure. I'm not sure if Professor Perry will touch on it or not. If she doesn't, which is totally fine, I might show you a couple of pictures. Right, there's been a lot of studies that are very definitive that show if you grow certain plants in the absence of fungi, they have a failure to thrive. If you add fungi right to that soil, it forms a relationship with the roots, and those plants grow to much greater, if we're talking about heights, right? And greater height, or just produces healthier plants. And so this is kind of a cool thing because it's showing the importance of this relationship, but it's also showing how, right, two invasive species can actually, in a sense, kind of work together and cause the spread, right? So, fungi is more than mushrooms. It's more than a pathogen, right? It is essential to the environment. And I think that's one of the things that this paper kind of starts to highlight for you guys a little bit. All right. Cool. So please, right, remember to look over those questions that I included to help you with these. Because, as you guys remember, some of these questions appear again on your exams. And for the most part, as a whole, as a class, you guys did really well on them, which is what I kind of expect. This is coming your way, and I want you to do well on it. All right. So with that being said, Lauren or Allie, who would like to go first?
I can go.
All right, let's do it, Allie.
Okay. So the article I read was called How Pandemics End, and it was posted in the New York Times. It was a bit of clickbait, to be honest, because I thought they were going to give an actual opinion of how they thought we should end COVID-19, but that's not exactly what they did. What the author said was there's really two ways that a pandemic or an epidemic can end. One of them is through medical or human intervention, and then the other way is just because people get tired of social distancing and they just learn to live with the disease. And then next, it talked about how fear plays a major part in the second way with the social ending, and gave an example of a man who was in Dublin. And he was a man of color. This was when Ebola was going around, and there was a lot of fear about people of color. For whatever reason, people just kind of associated him with Ebola. So he went into a hospital. He was actually dying of cancer, but he went into a hospital, and all of the doctors and nurses were too afraid to treat him because they were convinced he had Ebola, even though there were no reported cases anywhere in the area, and this was towards the end of the epidemic, so Ebola was really dying off. But the one doctor finally agreed to treat him, but it did come back that he was negative for Ebola, and so that just shows how much fear can play into pandemics as well. And then it gave the examples of the plague, which was in and out of history for around 2,000 years. And that's one example of a disease that ended through human intervention. And they're not really sure the exact cause of it. There are a few different hypotheses. They think maybe the cold weather killed the fleas that were living on mice. That's how the disease was spreading, and also through human contact. But so they thought maybe the cold was killing the fleas, or maybe that a different type of rat started carrying the disease. And then they also were burning villages where the plague was just really heavy in the area. And then they also thought another hypothesis was maybe that the strain just evolved over time to be less deadly. They're not exactly sure, but that's one example of an epidemic that ended through human intervention. And then another example is smallpox, but that was very unique because they were able to make a vaccine for it, and it's also not able to be carried through like animals and stuff. So if you could get rid of it in humans, you were rid of it for good. And then also had very obvious symptoms, so they could catch it quickly and isolate and treat people. But for an example of something that ended through just social aspects was influenza. So around World War One, when it finally had ended, the flu had kind of died down to a less deadly strain, but people were really ready for a new life and just to get on and be done with the war, be done with influenza. So they kind of just returned to life as normal. And even though the flu was still around, that was the end of a pandemic, just because people were just tired of living in fear and they just decided to live with it. And so at the end of the article, the author was just talking about COVID-19. So they were like, okay, well, what's going to happen in this case? Is it going to end medically or end socially? And really, there's not an exact answer, obviously, but it looks like it's leaning towards social because different states and areas are starting to reopen restaurants and gyms and public facilities. So it's looking like it might end through social aspect, but there's no telling for sure right now. And this article is important to the field of microbiology because the examples I gave in the different viruses, of course, would require knowledge of microbiology. So this is something that is affecting everybody in the world right now. And so it's really important that these scientists can figure out the causes of the coronavirus and, what the different results would be if we did open back up, how to treat people, how to get rid of it. And then for Christians specifically, we're impacted because not only church closures and social distancing, but because there's so many people living in fear that we're having an opportunity to open up the word of Christ and tell them, there's a God who loves you and cares for you, and worst come to worst, if you do die, there is a heaven you can spend eternity in if you just accept Jesus into your life. And then at the same time, it's just giving us an opportunity to be an example of what a Christian should be like, because so many people are living in this heightened state of emotion. So it's really important that we show them the love of Christ and show them that we care for them enough to take extra precautions, even if we don't want to wear masks or wear gloves or socially distance. We can show them that we care for them enough, especially the elderly and people who are immunocompromised. We can just show that we care for them enough to do that, even if, we don't really want to.
Cool. Any questions for Allie?
I have a question. Do you know if other countries are opening things back up, or is it just here?
It depends on the country. Like Sweden never really did big social distancing to begin with. They just told like the people who are elderly or immunocompromised to stay home, and everybody else for the most part lived as normal. But yeah, so it just depends country to country. Some places are doing what we are. I don't know of anybody who's on a super strict lockdown right now, but
I have just a question. When you're saying social, and I don't know if that's the term the author is using, so the author is arguing that we go out, or the social aspect would be going out and living our lives and letting the infection run its course, right?
yeah, that she did use the term social,
Okay.
and so that's kind of what she said. But for the flu example, it had already kind of died down to a less deadly strain anyways.
Right.
But I think she's trying to argue that in cases like the coronavirus that aren't as deadly and aren't like the plague, that people can just go back to normal and basically let it run its course.
Okay, interesting. Anybody else? All right, so, one of the things, and we can talk about this a little bit later, but I hope you appreciate is that pandemics are complicated, right? And, Allie referred to the one patient where he was in Ireland, I think, that they feared that he had Ebola. And so, even, and I didn't read a lot into this, but there was definitely at least a worry, right? If not some negative things that happened shortly after we found out the virus came out of China, right? And so some people call the COVID-19 virus, they called it the China virus, right? And so there was fear amongst Chinese Americans and even Asian Americans, right, that they would be associated, with the virus, right? And so, that can, be a real concern that maybe we don't think about when we label certain things. We'll actually talk about this. HIV had multiple kind of names early, and some of its early names or classifications targeted homosexual populations, right? And so there was a lot of fear of those groups of people and people outside of the community, right? And so there was people just assumed, well, if you're homosexual, you can spread the virus to me, right? And so there's a lot of things that go into a pandemic. And the last thing that I just want to touch on, and I like that Allie mentioned this, right, is there's two ways that this ends, right? And they both result in something called herd immunity, okay? And the ways that you get there, either you have a vaccine, right? And so you vaccinate enough of the population, right? So you basically trick the body into responding to the virus and building up protection. Or, right, like Allie was kind of mentioning, and this author seemed to kind of be hinting towards, right, this idea of a natural infection, right? Letting the virus run its course through the population. And after it's run its course through the population, theoretically, people are resistant to it, right? And, just to put it in perspective, vaccines typically take years to develop, to go through all the regulations and testing and procedures that they need to do. And the COVID-19 vaccine by a lot of people is being fast-tracked. And even a fast-tracked vaccine is 12 to 18 months from start to finish, assuming nothing goes wrong, right? And so that's still a long time. And so, I don't know where you stand, but it appears if it's not ready for 12 to 18 months, right, that there's probably going to be a lot of natural immunity that's built up as it runs through the population. So, Professor Perry, any other thoughts on that?
Well, I think I think you've explained it pretty well. I think it's just a matter of deciding how many people, are going to die if you let it do that, just run its course naturally. And if you're willing to make that sacrifice, because I was looking at a model last night about COVID, and they said if we were just to all go back now, stop the social distancing in Tennessee for totally stop right now, they would predict 35,000. I don't know how accurate that is, but within the next 3 months. That seems like a lot.
It does, right? And that's what makes this complicated, right? And I don't know. It's fascinating, right? Because there's a lot of things going on, right? You assume that everybody has sometimes the same education level or the same understanding, which not all people do, right? Sometimes it's hard for us to understand people that are maybe scraping by and need to potentially work to support themselves. But that doesn't mean that we want them to become infected and we want them to die. We don't understand, right? One of the things that's kind of scary about COVID is that we've seen some younger people that appear relatively healthy pass away from it. There's been some weird things that have gone on in some children that have had it. Now, these are still the minority of cases, but, right, it's a lot different if you're that one case or your family members that one case. And there's just still so much we don't, understand. And so it is kind of scary, right? Sorry, Emmeline was trying to join the conversation. Maybe later, all right, in a couple of years. And so it's a fascinating, kind of thing. And we're going to read a couple of COVID papers, and I'm going to kind of introduce it or kind of intersperse it amongst some of my lectures, right? Because we're living it now. And so there's a lot to consider in pandemics, right? And, the economy is one thing, as much as we hate to admit that, right? People's quality of life is affected by the economy. It's a really interesting thing. And historically, this will be my shameless plug. Next summer, assuming there's no pandemic, because it was supposed to happen this summer, but the trip got canceled. I do a trip with Professor Wood in humanities. And one of the courses that we do is history of disease. And we talk about a lot of pandemics. And, Ali mentioned the plague, right? And the plague actually changed life and the economy in a good way because it killed off so many rich people and upper class people in Europe that it allowed the lower class to move up and actually fill in some of those jobs and places in society. And so that is also not me saying that that's a good thing for us, but I think one thing that I hope we're hopeful for as Christians, especially, is that maybe there are some good things and positive changes that will come out of, I think, the terrible effects of this plague, right? So it would be really interesting to see what happens in the future. And I know we're taking way too long, but that's okay because it's fun to talk about it and it's fascinating stuff. Lauren, wow me.
Oh, geez. Okay. So my article was called Microbes Living in Deep Sea Rocks Spawn More Hope for Life on Mars. And basically it's about the ability of microbes to live in like really difficult environments. And so in this case, it was in the Earth's crust beneath the South Pacific Ocean, which is one of like the deepest parts of the ocean, apparently, according to Google. So like science knew that there were microbes that live in really deep parts of the ocean near underwater volcanoes because the volcanoes would cough up like rocks and stuff from the Earth's core, well, not the Earth's core, but deeper in the Earth. And they were mineral rich and they were really hot, so they would react with the cold water. And the microbes that lived around the volcanoes could use those reactions to get their energy. But what was significant about this paper is that these microbes they found were way farther away from the volcanoes than they thought were possible. And so they were drilling and they found them in the crevices of these really, really, really old parts of the Earth's crust. And so they aren't really sure how they survive there, but they think that the microbes use the clay that's in the crevices with them to get their energy. They think the clay is either made of like decomposing remains of other marine life forms that, floated down to the bottom of the earth as they were decomposing. And so they think it's either that or it's the Earth's crust itself, which is thought to break down over time. But they really aren't positive how they are surviving there. And then they found another kind of microbe that uses methane to survive and get its energy, but they have no idea where the methane comes from. So it's just a pretty cool finding, and they're just really have no idea how it works. So, so this article said this was significant because the environment that these microbes are in is very similar to the environment on Mars. They said that Mars has a wealth of methane, as well as rocks that are very similar to the chemical makeup of the rocks that these microbes lived in. And so, and in addition, there might have been an ocean on Mars at one point. And so they think that it really increases the likelihood that there could be life on Mars, which is pretty cool. But I think that this article is important to microbiology because whenever we discover a new organism, it raises the possibility of either using that organism or like a component of it or a chemical process that it has to solve a problem that we haven't yet been able to solve. And so that's why I think it's so important for new things to be studied, because it can help us develop new technologies, hopefully. And so I think that this affects us as Christians because it really reminds us of how much greater God is than anything we can understand. And I think that that's really vital to our relationship with God, because it's one of the reasons that he's so worthy of our faith and our submission, because I mean, he's so much bigger than we are. And so, you know, if he was something that we could understand, that begs the question of, is he worthy to be God? So when we find new things that we didn't think were possible, it really reminds us of how great he is. So
Cool. Any questions?
So I was just curious. You said that in the ocean there's this little like old like crevices. Do you know how like the bacteria that or the microbes that they found there, like how old they were? Like could they like how far they traced them, like how long they were there for, or like?
yeah, they just said that the crust was millions of years old. And so, I mean, they don't really know, I guess. It didn't say, but if the microbes are millions of years old as well, or if they came at a later point. But
Did they mention if these microbes produce any type of gases as their kind of metabolic byproducts?
no, they didn't say.
No, okay. I just asked because those of you that are interested in Mars, which again, I don't know how many of you that would be, is maybe none of you, but maybe you've heard this idea of terraforming, right? So the idea would be, could you change a planet's atmosphere into something that was more conducive to life, like for us? And so, I mean, this is all theory, right? But the idea would be that you could take a bunch of bacterial populations and even a bunch of different fungi, right? Because they can do some kind of similar things, and basically spread them over Mars or start these huge colonies of these things to start to produce gases that would then go up into the atmosphere, and theoretically you could change the atmosphere of the planet, right? But that would take like a long time. But that's why I was just kind of curious, you know, if they're like, this bacteria can survive on Mars, would it be beneficial to us? Any other questions? Should we sign Lauren up for the new Space Force that the country's getting, so we can send her out there with those bacteria and send her to Mars? Perfect. All right, cool. So I like that, right? That's kind of cool. Two cool variations. And so I love pandemic because that's totally relevant and going on right now. And I love Lawrence because it's microbiology that's really kind of fascinating and interesting, but it's not necessarily pathogenic, which I think we lose sight of a lot. So that was a cool variety of articles. So, and you guys have both, if you haven't, right, make sure you submit the papers along with like a link to your article. You can put that on the paper into Dropbox. Okay.
I just
Cool.
the title and author, is that okay, or do you want me to put the actual link?
Title and author, if you put that on there, that's fine. I can work with that. Okay. All right. With that being said, let's go ahead and get started talking about some genetics for a little bit, and then we'll start this, and then we'll take a short break, and then we'll finish up on it. So share 7. All right. So, this, and some of you have already had this, and so it will be review. I will throw out some kind of important differences between eukaryotic cells and prokaryotic cells that maybe you haven't heard of before. But again, the basic kind of function and structure of a lot of these nucleic acids and how we utilize them is very similar in prokaryotic cells and eukaryotic cells. All right. So just a couple terms, right, that maybe you know of or you've heard before, right? So genome, the complete set of genetic information. So when we say like your gut microbiome, we're talking about the complete set of genetic information that exists amongst all those bacteria that populate your gut, right? Same idea with the skin biome, your lung biome, your urogenital biome, right? So that's kind of what that's referring to, right? All the genetic information that's available, right? A gene, right, is a functional unit. So it's going to lead to the production of a protein, or in some cases, right, an enzyme, which is a specialized protein, right? DNA replication is talking about replication or duplication of chromosomes. Gene expression, right? When we say that, that means we're going from DNA to protein, right? In order to get there, though, first we have to go through transcription, which is DNA to RNA, and then translation, which is RNA to protein, right? And again, we'll kind of revisit some of these concepts and try to focus in or hone in on what's different between a prokaryotic cell and a eukaryotic cell, right? How are they doing it differently? One of the things that I want to go back to during this lecture is I want you to remember, right, that growth chart that we did with bacteria, all right? Let's see, here's my draw thing. Yeah, we'll use this. So if you remember, right, you had, if this was your work. My drawing thing's not working. All right, that's fine. But remember in the growth chart, right, you had the lag phase, you had the log phase, stationary, death, and then prolonged decline, right? And so what I really want you to kind of focus on is think about those two lag and log phases, right? So we'll actually talk about a little bit later. We'll give a good example of what's happening with enzyme production, and then what's happening with that growth phase, right? So that exponential growth, that log growth, how are they able to grow so quickly, right? And we can't grow at that speed. And so we'll see a lot of the reasons they can grow so quickly actually first start to appear or pop up in this chapter. And I'm sorry, my mouse has disappeared, so I'm trying to, well, I can't see it, maybe you can. Oh, let's see. Ha, I'm back. All right, good. A minor setback. Oh yeah, whatever. You accept this. And this is old. All right. So we're not going to spend a lot of time on this, but this is my boy Gregor Mendel. If you guys have ever done a Punnett square before in your past, it's from Gregor, right? He basically came up, with the idea of dominant recessive traits and used pea plants to figure those different things out, right? What was cool about him is he's a monk. Oh, Oh, it just came, it's white. That's why I can't see it. Why would it do that? Such a, ugh. Yeah, what are we gonna do? Anyway, and so I love this example of him, and you'll see this throughout various points throughout history that some of the best scientists were actually Christians, right? And so that the two, don't have to be separate. If any of you have heard of Francis Collins, who is the head of the Human Genome Project and is a big wig in the NIH, right? He's a professing Christian, so you can have it all, I guess. This is the basic dogma, and this was kind of on that first slide. So we say the dogma of protein, and basically you have this idea of you start with DNA, you then make RNA, you then make protein. And we'll briefly touch on ribosomal RNA, right, which is an important component of the ribosome, which we've talked about before. We also have transfer RNA, which I did mention in our seal finger disease. So tetracycline, right, interferes with the function of transfer RNA. And this is what's bringing amino acids to the ribosomes. But what we're really going to focus on the most in this unit is messenger mRNA. So if you casually are talking to your friends about RNA, which I don't know if you do or not, I'm guessing not, but if you were, this is probably the RNA that you're referring to as this messenger RNA, and it's going to result in the production of protein. Okay. So a couple different things, right? When we're making proteins from DNA, is that you have kind of a couple different options, right, that are going on, right? You have the ability to turn different genes on or off, even if they're located right next to one another, right? Some products of genes, you may need a lot of protein or a lot of enzyme, and you need it continuously, and so it's always being transcribed. In some cases, you may not need any, so you don't transcribe it, or you may need just low levels, and so you just transcribe what you need, right? And so our cells and the cells of bacteria are very efficient at spending energy where energy needs to be spent. A lot of genes, if you're wondering what was an example of something that's continuously being transcribed, well, that would be genes that are for the enzymes of glycolysis, right? Glycolysis is a vital process in our life that allows us to break down sugars into energy that we need. And so you always want those enzymes present or ready to go. All right, so that'd be an example of something that's always on. So again, DNA, mRNA, protein. What are the different things that proteins do? This is just a reminder, right? They can make enzymes. They can be used for molecular transport, right? Transmembrane proteins. We talked about flagella in bacteria, but they also play an important role in movement in our cells, even though we don't have flagella, with the exception of sperm, provide structure, right? Serve as sensors or receptors on the surface, right? And then gene expression bind to DNA. So this would actually be specialized enzymes, typically, that are helping translate and transcribe. Now, we're not going to spend a lot of time on this. You guys have seen this probably before at some point. Oh, there's one of my last script things here. Sorry, erase that. Not a great color choice by Zoom there. This is DNA. I just want to point out a couple of things. You can see a 5' end and a 3' end and a 5' end and a 3' end. We'll talk about why, all right, or how we distinguish which end. Actually, here we can see it on this slide, so that's fine. So here you see the 5'. There's actually a carbon right here, and this little bend in this line. This is a carbon. This is actually the 5th carbon, right? This is the 4th carbon, and here, right, is the 3rd carbon. So that's how they get their 5', 3' end. They're going opposite directions. So if you've heard of DNA being anti-parallel, right, that's because, right, they run in the opposite directions. Here you can see the base pairs, right? So AT, GC, we'll talk about those in just a second. And you can see it forms, right? We call this an alpha helix, right? So that's kind of that twisting or bending, right? So this contains all of the information that we need to make all of the proteins that we need for life. Okay. This is the basic structure of a nucleic acid, right? RNA and DNA are almost identical. They're very similar. So here you have a phosphate group. Here you have your 5-carbon sugar, right? And then here you have a nitrogenous base. All right, so when we talk about DNA and RNA, the biggest variation is going to exist in the base, right? So this kind of side structure here. This is just reminding you of counting those carbons, right? So here's the 3' side, here's the 5' side, right? So the nucleotide that this would be bound to would be going in the opposite direction, right? Here are your different bases. So you have 2 categories, right? You have your pyrimidines and you have your purines. So cytosine, uracil, only in RNA, thymine, only in DNA, okay? And then you have adenine and you have guanine. Notice besides structural differences, or the structural differences lead to differences in the number of hydrogen bonds that exist, right? So adenine and thymine, right, have 2 hydrogen bonds. Guanine and cytosine have 3 hydrogen bonds between them, all right? So which base pairing is stronger?
GC.
GC, right? It's just an additional hydrogen bond. And one of the things, and I think you guys did pretty well on this in the test, is individual hydrogen bond, right, is relatively weak. When you start to stack them on top of each other, though, it becomes very strong. And so if you think about a big strand of DNA, there's a lot of hydrogen bonds in there, and so it actually becomes a relatively strong bond, right? So this is kind of our first look at an important difference in the replication of DNA between prokaryotic cells and eukaryotic cells. So if you remember, from a prokaryotic cell, it has a circular chromosome, right? And the circular chromosome has one origin of replication. What that means is that the enzymes that are going to replicate DNA bind to this one spot, right? Within your DNA, you have multiple origins of replication, which means you have to have multiple enzymes binding to these different spots in order to replicate your DNA. They have one site. And because they have a circular chromosome, they, right, actually replicate in both directions. So we call these replication forks. Okay? So if you think about it, if you have to make a copy of something, right, and you have the same length, is it quicker to just work your way down? Or is it quicker to start at each end and work your way towards the middle? Right? The answer is, I hope you're thinking in your heads, is it's work your way towards the middle, okay? And so this is going to increase the speed with which replication can take place. Think about it. First of all, they only have one chromosome. How many chromosomes do you have? Let's see. Sarah, how many chromosomes do you have?
Sorry.
That's okay.
Unmuted. We have 46.
46, right, or 23 pairs, right? So you're making copies of all of those chromosomes every time one of your cells divide. They have one, right? Now it's big, but they have one, right? They have one chromosome, okay? And that one chromosome that they have, right, they are going in both directions to replicate it. So start to think, right? Start to think. That exponential growth in the growth curve. This is one of the reasons, right, that they're replicating so much more quickly than our cells. All right. So just kind of try to keep that in mind as we go along. In case you're wondering too, if you see this term semi-conservative, you can see in the 2 chromosomes that one of them has the original strand, right? So that's the dark purple, and one of them has a new strand. And so, right, each copy, right, has an old section and a new section. All right. So kind of keep that in mind if you see that term. Okay. So this is what I already mentioned, right? Proteins bind to the origin of replication. We were just focusing on a chromosome, but plasmids, it's the same concept, right? One origin of replication, plasmids are circular, and so you can copy those relatively quickly. Eukaryotic cells, right? Multiple binding sites, right? Multiple origins of replication in order to get a duplicate of our DNA, right? Now, one of the things that we need to keep in mind and right, the process in prokaryotic cells and eukaryotic cells is really similar, is that prokaryotic DNA, eukaryotic DNA is tightly wound, all right? So they wind around nucleoid-associated proteins. We wrap around histones. In order to replicate, right, DNA, or even in order to transcribe DNA into RNA, you need to unwind it, all right? And so you have DNA gyrase and DNA helicases, which are going to essentially break and unwind DNA. And when they break and unwind DNA, it actually opens up a spot for something called primers, right? Primers are basically little place fillers that exist, right, in the exposed strands of DNA that allow the DNA polymerase to lay down new base pairs as you make that copy of DNA. Okay. And so here you can kind of see this process occurring. And so please note that replication proceeds more quickly when you are building in the 5' to the 3' direction. That means your DNA template strand, right, is going the opposite direction. You can go the other way. We'll talk about that briefly. If you've heard of something called a lag strand, right? That's actually going the opposite direction on the other strand of DNA. So please note it doesn't show it here, but somewhere over here, right, is another red strand, right, the original DNA, and you are making copies of it also, right? So you're gonna have 2 strands. So here you can see the DNA polymerase. It's laying down new base pairs as it follows the directions, right, of this template strand. This is kind of a nice little figure. I know there's a lot going on here, but it's kind of showing what this looks like. So here again, we're building 5' to 3'. So this is the leading strand. The lagging strand, you're basically going the opposite direction, and you're building that in small chunks, right? So those are called Okazaki fragments, if you've heard of those. And so the leading strand is done much more quickly than the lagging strand. However, right, regardless of that, right, you see that you are ending up, right, with this again, that's that semi-conservative. So red's the original strand, blue is the new. And as this goes through, right, you see the helicase unwinding this so that you can make these copies. All right, and again, we're not going to spend a lot of time on that. We can certainly talk about that if you'd like to via email or a Zoom session, right? Or if you have some more questions, right? If you want to know a little bit more, you can, dive into your textbook. But I'm just kind of trying to gloss over the basics here for you guys. All right, so let's talk about RNA. So RNA, you actually use only one strand of DNA as the template. So remember DNA, right, that anti-parallel, it has 2 strands. only one of those strands is actually a template, right, or a coding strand. And from that, we get an RNA transcript. RNA, unlike DNA, is single-stranded in prokaryotic cells and eukaryotic cells. We will mention, or at least talk about, right, some viruses actually have double-stranded RNA, right? Viruses kind of throw a lot of these genetic principles or concepts out the window, right? They have some unique features that we'll get to. I already mentioned 3 major types of RNA: messenger RNA, ribosomal RNA, transfer RNA. Again, we're going to spend the majority of our time referring to messenger RNA because that's what's going to lead to the production of proteins, all right? So for prokaryotic cells, right, they have something called a sigma factor that is going to help direct RNA polymerase, or to help direct RNA polymerase binding to DNA, right? So DNA polymerase, right, makes copies, right? It replicates DNA. RNA polymerase binds to DNA to then make RNA, right, from the DNA strand, all right, or using it as a template strand. And so the sigma factor helps direct this RNA polymerase, okay? So sigma factor directs RNA polymerase where to bind. It splits the DNA. RNA polymerase adds nucleotides. Remember, we no longer have thymine, right? We have uracil. RNA polymerase links these nucleotides, and then the hydrogen bonds between RNA and DNA are broken, right? And you have to break these bonds because RNA is single-stranded, right? The DNA then fuses back together with this complementary strand of DNA. And sigma factors, right, are important to point out. If maybe you guys have had some genetics background before, talked about, if you've heard of something called a transcription factor. Transcription factor, right, is what helps RNA polymerase bind to eukaryotic cell DNA. A sigma factor is the prokaryotic equivalent, right? We'll talk about those a little bit more. Again, this is a nice little summary here. Here you see negative strand, positive strand. This is referring to DNA. And so it's the negative strand that serves as the template for making RNA, all right? So that negative strand, you'd have all the nucleotides, the RNA would bind, right, the matching nucleotides to it, and that's what you then break apart, and the RNA goes on its way. The positive strand, right, is complementary to the RNA strand that you've just made, and you could actually look at the positive strand and immediately know the RNA sequence. The only difference is you have to change out uracils, or you have to change out thymines and put in uracils for them. Promoter, right? This is where RNA polymerase is binding. So this is what the sigma factor directs the RNA polymerase to. Right? So here's the sigma factor. It mentions it down here. And then promoter is where we start. Terminator is where we end. And we can actually see that right here. Okay. So here's a strand of DNA. Here's the promoter region. The promoter region is a series of nucleotides that the sigma factor recognizes, okay? Here you have the terminator. So once you go down this sequence of DNA, you get to the terminator and you stop production right there, okay? So let's just walk through this really briefly. So here's the RNA polymerase. It's directed to the promoter by the sigma factor. Right, RNA polymerase starts to move down the DNA. So notice this red one. This is the negative strand. We're using it as the template. Okay, and as this moves down, we see the RNA, right, starts to kind of lag behind it. It's kind of like a string, right? So as you make RNA, right, you release it and you start to see it kind of trailing behind. When the RNA polymerase gets to the terminator, it comes off, and then you have your RNA transcripts. Okay. What we're going to do is we're going to pick up right back here. It is 9:20, so let's take a 10-minute break to 9:30, and we'll finish off at least chapter 7. We might jump into chapter 8 very briefly. Okay. Okay. Yep. I can do that. All right. So let's take a break. 9:31 is when I want you guys back, and we'll go from there. All right. Resume recording. All right. So what we have here is basically the exact same slide as before. But what I wanted to emphasize on this is that in prokaryotic cells, what we see happen is that as soon as an RNA polymerase has moved off of the promoter region, another RNA polymerase is able to immediately bind and start making the same transcript, right? So you can have multiple transcripts that are being made at the same time. We don't necessarily see this in eukaryotic cells, right? And so in a eukaryotic cell, typically, right, if you want multiple transcripts, RNA polymerase binds, makes its transcript, another RNA polymerase binds, makes its transcript. That's not the case in prokaryotic cells, right? We see multiple transcripts being made at the same time. Again, think about that exponential growth or that log phase. How are they able to grow and divide, right, so quickly? Well, I guess grow would be the same as dividing them, right? But how are they able to divide so quickly, right? How does their population grow exponentially, right? And this is one of the reasons. So now we have replication, right? We have multiple transcripts being formed because as soon as an RNA polymerase clears the promoter, another one can bind to the promoter. All right, so keep that in mind. And you know, this is just an important time to kind of point out gene expression, right? So that RNA that we are making, right, if it's messenger RNA, which again is going to be the majority of the RNA we're talking about, will eventually become, right, a protein. This is where I wanted to introduce this idea of monocistronic and polycistronic. All right, so oftentimes, all right, one thing I want you to realize is that to perform a certain function or a task, you're probably going to have the contribution of more than one protein. In eukaryotic cells, right, we're mainly monocistronic. And that means, right, that let's say you need 3 proteins for a certain function. Well, here you have the promoter, right? You have your transcription, right, region. So you make this little bit of RNA here. You have the terminator, it pops off. Somewhere further downstream, perhaps, you have another promoter, you make a transcript, there's a terminator, it stops. Further downstream from that, you have another, right? And so they're kind of spaced out. So one at a time, right? One gene at a time. Polycistronic, which we'll actually see a great example of here in not too long, is basically when you have separate genes that may be working together, right, to perform a similar function that are lined up back to back to back. And so in one transcript, right, you were actually getting the product of 3 different genes, as an example, right? It could be 2. Also, right, poly just meaning many. So you'll see what that looks like here in just a little bit. All right, again, translation, right? So we've made our transcript, we've made our mRNA, so we have that here. Ribosomes, right, which are composed of ribosomal RNA and protein are important, and then that transferred tRNA that I mentioned before is also important, and you'll see how those are coming into play in just a second. When it comes to translating, right, so think of translating as reading. So you've made that mRNA, you now have to read it, right? You read those instructions, and when you read those instructions, that's how you get protein. For RNA, for mRNA, the instructions come in 3 nucleotide bursts. We call those codons, all right? So every 3 nucleotides is a codon, and it tells us something. One of the codons that every transcript has to start with is the start codon, right? This is AUG, right? All proteins start with methionine. Now, a little cool difference, and I think I have this somewhere later. Prokaryotic cells actually have formal methionine, and you'll see how that's spelled in just a little bit. But it's a slight modification to the methionine amino acid. But just note, right? It's a modification. It's still methionine. Everything starts with methionine, no matter what you are. Right? You also have a stop codon. All right. And for the stop codon, you actually have 3 different possibilities, right? UAA, UAG, and UGA, right? Go Dogs. I don't know if they're not really, they always disappoint in sports, but at least they don't disappoint in stop codons. Right, Noel? Right. Good. All right. The area between the start and the stop codon, right, is called the open reading frame. All right. So this is where, right, the majority of that protein that you're building, right, the majority of the information you need for it is going to be housed. Now, you will sometimes see that the codons referred to as degenerate. And degenerate, right, is not like a degenerate in society. Degenerate is actually referring to this, the fact that you have multiple codons that are coding for the same amino acid. If you've never seen a chart like this, it's kind of fun because you'd be like, okay, what happens if the first letter of the codon is C? And then you want to say, what happens if the second letter is A, right? And then the third letter is U, right? And so then you can kind of, that's how you kind of follow that chart. And here you can see, right, in some cases, you have up to 4 codons that give you the same amino acid. So that's what it's referring to as degenerate, right? Multiple possibilities to give you the same thing, right? The exception being, right, that methionine right here only has 1 possibility, right? So there's no kind of variation there. You see some have 2, some have 3, right? Some can have 4. That's what degenerate means. So please keep that in mind if there's a question about that, or if you see the term degenerate, right? It's referring to multiple codons for the same amino acid. And so this is kind of what this looks like. So in all these cases, right, it's showing you open reading frames. And so open reading frames is every 3 nucleotides. And so here you can see, right, 3 nucleotides, 3 nucleotides. So amino acid, amino acid, amino acid. And as this mRNA transcript moves through the ribosome, right, it's translated, and the codon's read, right? And then these are the amino acids that are stuck together to help form the protein. The thing that's nice about this is it shows you the effects of shifting the reading frame by individual nucleotides. So up here you see CUG, GGCA. So what they've done down here is they've said, what happens if we shift over 1 nucleotide, right? And so now we have UGG, right? So if you did UGG, right, so you kind of started reading at this U instead of this C, you can see it gives you a totally different set or arrangement of amino acids. And I want you to file that one back away because that's going to be important when we actually talk about mutations a little bit. Does anyone have any questions on codons or mRNA? I realize that some of you that maybe haven't had this before, this can kind of be a lot to digest. And so I really want you to have the chance to let it sink in. Some of you that have had this is probably just scratching the surface, which is totally fine also. So if there is a question, go ahead and shoot me something in the chat box. Sometimes I don't see the chat box, so just go ahead and yell at me through Zoom. That's fine too. Oops, actually, let me go back there. So this is the transfer RNA. Again, we don't talk about it much. You'll see it one more time. But here, right, you can see the mRNA transcript, 3 nucleotides. So this is the codon. Transfer RNA contains something called, what do you know, the anticodon, which binds to the codon, all right? So each codon has an anticodon that's carrying, right, a specific amino acid, right? So in this case, proline. And so When this codon is read by this anticodon, it brings in proline, right? These three nucleotides, presumably, right, are, well, they're going to be another amino acid. It might be the same, it might be different. And it's going to come in and then bind to this proline that this transfer RNA brought in. And you'll see this in a diagram here in just a second. How does transfer RNA get its amino acid? This is kind of random. I don't know if this has ever worked to tease this answer out of you, but one of the things that I want you to appreciate, right, aminoacyl tRNA synthase, I'm not going to ask you to memorize that, but what I do want you to appreciate is, again, we've mentioned this, enzymes. Enzymes are directing almost every aspect of cellular function, right? And so here, right, DNA polymerase enzyme, RNA polymerase enzyme, helicase, ligase that are helping unwind and break apart DNA, enzymes, right? And so you just can't get rid of them. Enzymes. All right. So here's what translation looks like. And again, I'm not expecting you guys to be translation masters, but I do want you to kind of appreciate a few things here. Here's your RNA transcript. Every 3 nucleotides is a codon. Here you can see your ribosome, right? If we're talking about a prokaryotic cell, this top orange one is the 50S subunit. This bottom kind of reddish one is the, what subunit is it? The 50 and the what? Can you remind me, Lauren, what's that smaller one? This is a 50S. What is this one?
Hello? Am I unmuted now?
Yeah, you're unmuted.
Okay, is it the 30?
30S. All right, good. 30S, right? If we are talking about, Lauren, I'll just keep you on while you're unmuted, all right? We're talking about a eukaryotic ribosome. What would the size of the 2 different subunits be?
40 and 60.
40 and 60, right? Perfect. Thank you, Lauren. All right, so your 50S, your 30S, you'll see when we get to the antibiotic unit that different antibiotics are interfering with different components of these units. And remember, tetracycline that we mentioned in the seal finger disease is interrupting the ability of transfer RNA to interact with the ribosome. If transfer RNA can't interact with the ribosome, then we can't bring in amino acids and we can't synthesize proteins, right? So here you see kind of 2 different sites. You see the E site, the P site, and the A site, all right? The way that you can kind of look at these is the P site is basically kind of where the magic starts to happen, all right? The A site you kind of think of almost as like a little bit of a holding site, and the E site is the exit site. So you have methionine, right, because it's the start. It binds in the A site. You then have, in this case, proline comes in. When proline moves into that holding area, these 2 connect to one another, right? They'll then shift over. So the transfer RNA that brought methionine in will enter the E site. It will leave. The transfer RNA that brought proline in will wait in the P site. Something else comes in the A site, and you just start building and then moving transfer RNAs out. Transfer RNAs, in case you were wondering, can be recycled, right? They can go and pick up other amino acids and then come back. So let's continue to highlight then some of the differences between prokaryotic cells and eukaryotic cells. And one thing, I want to go back here and I'll do this here because I know I mentioned it in the text, and I'll probably show it again anyway. Another kind of cool feature of prokaryotic cells is that, again, as soon as this start codon moves through this ribosome, another ribosome can move on and start another bit of translation immediately. So you have multiple transcripts that are coming off. You then have multiple translations occurring of those same transcripts. All right, so again, the name of the game is exponential growth. Multiple transcripts, multiple translations of those transcripts. And so we're talking about proteins and we're talking about enzymes, right? Remember, every enzyme is a protein, not every protein is an enzyme. You're able to make those very quickly. If you're able to make those very quickly, that also means that you're able to replicate and duplicate yourself very quickly, right? Think about exponential growth. What's going on in that growth curve? And that's what we see here, all right?
I have a question.
I have an answer.
Okay, so I
Evan.
know after a ribosome like translates part of an RNA, it dissociates the each subunit. Is a ribosome, can like any two subunits come together, or is it like the same two subunits?
I've had to look specifically, but as far as I know, as long as it's a 50, like we're talking about prokaryotic cell, as long as it's a 50S and it's a 30S, right? As long as a 50S has a 30S, it doesn't matter which 30S it is, because there shouldn't be variation in those subunits, right? I would have to double check, but I would almost be willing to bet your life on that.
That's not nice.
You know what? I will. No, I'm just kidding. But yes, as far as I know, and I can officially double check, but those should be interchangeable because there shouldn't be a variation amongst them. Okay, good question.
Okay, cool.
All right. So these are some things that I want you to kind of think about, and some of them, in my opinion, will be, kind of I'll say an aha moment. It's not overly complicated, but maybe you haven't thought of it before. So for prokaryotic cells, if we're talking about transcription and translation, these are all taking place in the cytoplasm because there's no nucleus. Right. In eukaryotic cells, you can come down here and see this point. A transcript, right, is made in the nucleus. It then has to be sent out of the nucleus, where it's translated by ribosomes in the cytoplasm or ribosomes in the rough ER. Those of you that remember, right, your basic cell structure of a eukaryotic cell, remember that the nucleus, right, is a double membrane, has a double membrane around it, all right? It's called the nuclear envelope, and within the nuclear envelope, you have nuclear pores, right? And so that allows these transcripts to move out. And so you have to make a transcript, you then have to send it out. In a prokaryotic cell, everything takes place in the cytoplasm. It's all right there. Things don't have to move around nearly as much, right? Translation begins as transcription is still occurring. So if you remember when we're making our mRNA and you kind of have that tail of it hanging off, right, as it's finishing, as soon as there's essentially enough RNA to grab onto, the ribosome can attach and be translating while the transcript is still being finished. All right, so very, very quick. We can't do this because we have to wait for our transcript to be sent out for it to find a ribosome. All right. Again, not a huge difference, right? But this is the formal methionine that I mentioned, structurally slightly different for the start codon for prokaryotic cells. However, right, we actually do have this present in us, right? We find this in the mitochondria, which we've already kind of mentioned as being kind of bacteria-like. And we also see this in the chloroplast of plants, right? Our start codon is just regular methionine, right? Again, I mentioned this multiple ribosomes, right? Polyribosomes. So you can be translating, multiple ribosomes can be translating the same strand of mRNA, right? So very quick. And so that doesn't happen here, right? We have one ribosome translating, it finishes translation, and then another one can begin. So again, this is all contributing to the speed. One of the things that we didn't mention that you can kind of pull away from here too is, I mentioned in DNA replication, right? They have one chromosome. If we're talking about us, right? We have 23 pairs or 46 total. Another thing, right, with mitosis, those of you that remember mitosis, one of the first parts of mitosis is that nuclear envelope has to dissolve or disintegrate before you can start pulling things to the different side of the cell to get your copies on each side. A prokaryotic cell doesn't have to have a nucleus dissolve before replication can occur. So when you talk about replication, one chromosome, you don't need a nucleus to dissolve in prokaryotic cells, right? And that one chromosome, right, that bidirectional replication. And so think about it, replication can happen more quickly. Transcription and translation can happen more quickly, all contributing to that exponential growth. Please, right, kind of try to tie those together. If you're just trying to learn the difference between transcription and translation, I don't really think that will stick for all of you. It may stick for some of you, but if you're associating them with that exponential growth, knowing why bacteria grow so much more quickly than we do, I think it will help, right? It gives it a reason, or it gives it context. All right, something else. Let's add to the speed. All right, post-translational modification is something that occurs with proteins. So often proteins still need to be folded. Sometimes we have chaperones that aid in the folding of these proteins. All right. We also have our signal sequences. So this actually, let me take a step back. This doesn't necessarily directly have to do with speed. I got one slide ahead of myself, but this is still important, right? So proteins have three-dimensional structure. They need help folding. Signal sequence, right? You guys saw really early on when we talked about digestion of macromolecules by bacteria, that one enzyme was able to move out of or across the cell membrane because it had a signal sequence, right? It knew where it needed to go, right? We also see the addition of sugars and lipids to proteins, which is really normal and contributes to function, right? In our cells, the addition of these sugars and lipids is taking place in the endoplasmic reticulum. Prokaryotic cells don't have an endoplasmic reticulum, and so this is happening again within the cytoplasm. All right. So let's actually talk about, and maybe I'll think about rearranging this in the future and putting this with transcription. For eukaryotic cells, we produce something called pre-mRNA. And basically, our pre-mRNA cannot be processed, cannot be translated by the ribosome until, all right, it's capped with methylated guanine. We add a poly-A tail, right, adenine derivatives, and we splice introns out, and we put the exons together. In your case, you're like, what is he talking about? Well, here's an example, right? So this is eukaryotic DNA. Okay. So in eukaryotic DNA, here you have exons and you have introns, right? Think about introns as intruders, right? They are a sequence of DNA that does not have anything to do directly with gene function, so we need them out of the way. So we transcribe our DNA into RNA, pre-mRNA. We add our cap, right? Our methylated guanine cap. We add our poly-A tail, right? We then splice out our introns. We push together the exons, which are the segments of the gene that we need, and then we translate it. All right, so our RNA has to be processed before translation. Prokaryotic cells, you probably guessed it. It's different, and here's how it's different, right? Prokaryotic cells, the mRNA is not processed. We don't have to add a methylated guanine cap. We don't have to add a poly-A tail. There are no introns, right? So we don't have to cut anything out and splice it back together. And then we already kind of talked about some of this, right? Translation of mRNA in a prokaryotic cell can begin as we're making transcripts, right? Here we already mentioned the transcript has to be sent out of the nucleus before it can be translated by eukaryotic cells. We mentioned polycistronic, right? Multiple genes, monocistronic for eukaryotic cells, one gene at a time. Okay, so this is kind of a good summary table. And again, we've talked about these kind of bottom 2 points, but this point can be kind of added again. Think about that exponential growth. We know differences in replication. We know differences in transcription, translation, and you can throw in there how the mRNA is being processed. Essentially, in a prokaryotic cell, it doesn't have to be processed. As soon as you make that transcript, it's ready to go. That's not the case in eukaryotic cells. There are different things that you have to do to it before it gets translated. All right. So this is kind of just going into some basics. You know, why are genes expressed? What triggers their expression? And we kind of have two basic categories, right? We have steady state. And so one of the things to think about in here is metabolism. So GAPDH is an important enzyme in glycolysis. Glycolysis is important for breaking down sugar and harvesting energy. And so I already kind of mentioned this, I think maybe yesterday. I don't know, they all blend together, right? It was probably today, though, now that I think about it. But you want these genes constitutively activated, right? That means they're always on. You're always making the products, whether proteins or enzymes, that you need for certain metabolic process. They're so important, right? If they're important for harvesting energy, you probably want them on almost all the time, right? Other things could be environmental changes, right? This could be, in our case, external or internal, all right? For single cells, right? There's not, like prokaryotic cells, there's not necessarily a difference there. It's all kind of external for them, right? But it could be some type of stress or danger, right? It could be a nutrient abundance, or it could be nutrient deprivation, right? All of those different things are going to signal to that cell that certain genes need to be expressed, right? So we can go back, relate it back to that iron paper, right? Remember, when iron is overly abundant in a system, we don't need, if we're a prokaryotic cell, right? When I say we, I'm referring to prokaryotic cells like I'm one of them, right? We don't need to express those genes that are great at stealing or sequestering iron because there's so much of it in the environment. However, when we are deprived of iron, then we turn that gene on, all right? And this idea, right, of responding to these stresses or these changes, it's really important for conserving energy, right? You don't want to invest time and ATP into producing a bunch of something that you might not need, okay? So this is an important concept in prokaryotic cells and eukaryotic cells. Good. All right. So let's talk about signaling, specifically this idea of signal transduction, right? Please don't get this confused with transduction of horizontal gene transfer, which we'll talk about at this point probably tomorrow. Actually, let me see. Oh, okay, we'll finish this up. So quorum sensing, right, is this idea that you hit a critical cell population or density, all right? So when you have enough cells present, they can release signals, and those signals can find a target. If you have two bacteria on the opposite side of a room and they're releasing signals, those signals probably won't find each other. However, once they start to divide and produce more bacteria, then that's going to be more targets for this signal. And quorum sensing is essential, right, in communication in biofilms. So we talked about biofilms, and we talked about, and you guys did pretty well on this on the exam, their ability, or cells' ability to communicate with one another within a biofilm. And quorum sensing is how they do this, right? So once a critical mass has been reached, right, a critical size of the population has been released, the signals, right, that these bacteria release, right, call these the bacteria's voice, right, can find a target, and they can communicate and send signals to one another and communicate that way. All right. So this is a slide that I got from Dr. Chrisman, and it's basically showing kind of what quorum sensing looks like. So quorum sensing signal molecules, right? So if you go back one slide, right, here you see the red, the blue, the green, right? So they're a product produced by that bacterial cell. They are released, right? They find a receptor or a target. And once they find that receptor or target, they bind or enter the cell, right? They cause transcription of a segment of DNA, right? That leads to the expression of some type of gene, all right? So that signal that they send out, it triggers the production of a gene product. And what's cool here too is that sometimes this signal can actually act on the very same cell that released it. Right? Those of you that have had anatomy and physiology before, and you've done the endocrine system, right? You've heard of the autocrine effect, right? A cell releases a signal, and that signal actually acts on the very same cell and leads to some type of response. So you can see both of those things happening. All right. Here, right, you see basically this concept, and this is not unique to prokaryotic cells, but it's important to kind of mention that membranes, we talked about this, right, are semi-permeable. Sometimes things can get across, sometimes things can't. And that's okay. Even if you can't get across, right, you can affect the signaling inside of a cell. And so this is just showing, here's your stimulus, your signaling molecule. This could be a signal from another bacteria. It binds to a receptor, right? And that receptor, right, has a response regulator, some type of protein in here. And when this stimulus binds, right, it signals an effect in that receptor, and you cause a cascade signaling effect. And this happens even in the absence of that signal entering the cell. All right, so it's a way for something from the outside to cause an internal effect, even if it can't cross the membrane. All right, so again, let's kind of talk about some of the important things that gene expression is contributing to as far as bacterial survival. All right, and what's really cool about gene expression as it relates to survival in bacteria is that sometimes, right, it can actually be random. And that randomness is really, really important. All right, so one of the ways that gene expression in a random kind of variation or assortment contributes to the survival of bacteria is through antigenic variation, right? Surface proteins. And so internally, the metabolic activities and functions of the prokaryotic cell aren't really changing, but externally, they may look a little different. So my example is here. This is my boy, Bill Murray, right? The 1984 classic, Ghostbusters. You know, this actually reminds me of a little bit of Allie's talk. One of the negative effects of pandemics is that I was looking forward to Ghostbusters Afterlife being released July 20th. It will now be released next year. So, you know, the pandemic is affecting many things, even my movie schedule. I mean, if I could have one thing that would remain normal, you think it'd be my movies, but that's fine. Deep breath, right? But here's Bill Murray, right? Totally different outfit, still Bill Murray, totally different outfit again, still Bill Murray. And so I use this kind of jokingly, but this is antigenic variation, right? He is still the same person, right? But he has different outfits on. Same idea kind of in bacteria, right? What they look like on the surface or what they express on the surface varies, right? And this is really important if we're talking about their interaction with our immune system. If our immune system is looking for Bill Murray dressed up as a Ghostbuster, but then he changes his outfit to Bill Murray from Life Aquatica to potentially after that Bill Murray in a suit, our immune system is smart and it'll figure out eventually, but by the time it takes for the immune system to figure it out, Bill Murray may have divided even more, all right? So it's a way that they can survive, right, or evade our immune system, right? It gives them a little bit of extra time, right? Phase variation, I like this. This is an on-off switch. And an example of phase variation is actually the expression of things like fimbriae that are keeping bacteria rooted or anchored. So if you remember, one of the important things about a biofilm is that as that biofilm grows, right, some bacteria are released, right? And when those bacteria are released, they can then find a new home. And in phase variation, right, bacteria will randomly have fimbriae that allow them to attach, randomly have fimbriae that make them let go. And this actually assures this kind of random expression that even if things are really, really good, some bacteria are constantly being released because sometimes things might not be good, all right? And then sometimes as those bacteria are released, we'll turn those fimbriae on and they'll grab onto whatever environment's near them. Some of them, right, won't find the right environment. Some of them will, all right? So this randomness, right, kind of ensures the survival of certain species, right? And so it's advantageous sometimes to be randomly turning genes on and off. It's an important survival mechanism for natural selection, right? Cool. Gene regulation, we've already kind of mentioned some of these, but we can put some fancier terms to them. Constitutive is always on. Again, examples, enzymes of the central metabolic pathways, right? Really important for harvesting energy, maybe really important for building things. We need them on. Inducible, all right? These are not routinely produced at significant levels. They are important. And so these are enzymes that are involved in the transportation or breakdown of specific nutrients, all right? We'll actually show an example of these, or of an inducible gene in just a little bit via the lac operon, all right? And then repressible, right? These are produced routinely. However, they can easily be turned off when they're not required, right? So some of those things that are building, because it's an energy investment, you need them a lot. But again, if the energy state of the cell is low, well, then we just turn them off and wait until we have energy to build things. So how, right, or what are some of the different ways that bacteria can regulate this or regulate their gene expression? Well, one regulation point is transcription, right? And so here you have alternative sigma factors, right? So that means for different genes, you have different sigma factors. And so if you need a gene on, you need to make sure that sigma factor is produced. If you don't want a gene on, then you can actually repress that sigma factor, all right? And so you have anti-sigma factors that can interfere with the ability of these sigma factors to bind. And sometimes I think we have this idea of thinking anti-sigma factors are bad because they're inhibiting a sigma factor from doing what it wants. That's not true. Anti-sigma factors are good because they're making sure bacteria don't waste their energy producing something they don't need, right? So you can basically determine, can RNA polymerase bind or can it, right, through regulation of these sigma factors. Now let's talk about some specific DNA binding proteins and how we kind of categorize these. So you can have repressors or you can have activators. A repressor, and we'll show examples of these in just a second. A repressor is going to, well, actually, you know what? Let's see. Got a lot going on there. That's chapter 8. I'm not going to get to that today. What's that? Hold on. Sorry, guys. Yeah. So this is what we'll do. We have 10 slides left here, which is not terrible. So we'll start with this tomorrow, and then we'll do chapter 8 and finish with mutations, and maybe even jump into a little bit of taxonomy. All right. So we'll start here. We'll start here on Wednesday, right? Because today's Tuesday. Today's Tuesday, right, Rachel? Perfect. Right. Talking about repressors and activators, because I do want to spend a little bit of time on the lac operon and catabolic repression. All right. So I'm going to stop here. It's 10:11. Let's just take, again, another brief 10-minute break. I'm going to sign off. I'll sign back on. We'll split into groups. I'll talk briefly about what your activities are going to be today, and we'll do some basic lab announcements then. All right. So get out of here for 10 minutes, walk around, go to the bathroom, get a drink, get a snack, do what you got to do, and then we'll hit the ground running with lab. Okay.